Hello and welcome to Nikolai's genetics lessons, and today I have prepared two questions for you. And the first question is, most genes in prokaryotes and eukaryotes are regulated primarily at which level of expression? And here are the 5 answers to choose from. Answer A, transcription; B, messenger RNA stability; C, RNA processing; and D, protein activation; and E, translation. So this is double-stranded DNA, and messenger RNA would be made from the template strand of the DNA, and it would consist of exons and introns. Of course, prokaryotes don't have introns. Then this messenger RNA would be used in order to make mature messenger RNA, and then ribosomes would attach and would make a protein. In many cases, this protein would be not active. Example would be insulin. It have also to be processed later. One part would be cut away, and we are going to get final enzyme. So this would be the first stage, which we call transcription. Then immature messenger RNA would be processed, and then translation and final modification of the protein. Once again, first answer transcription, then messenger RNA stability, because here messenger RNA would be processed, poly A tail would be added. If an example of eukaryotes, prokaryotes wouldn't have this processing machinery, and usually when messenger RNA would be produced, at the same time ribosomes would attach and start to produce protein, and then protein activation, translation of the protein. And I would use another example from the life. Imagine that you want to paint a room new color. So what would you do? You would take a car, would drive to the nearest store, you'll buy a paint, would come back home, you would take a brush and you would stir the paint. So this would be your steps. At which step it would be better to, say, cancel painting of the room? Of course, it's the first stage, because with every stage, with every step, you are spending your resources, such as money and time. And the same is true when we talk about the processes that going on inside the cell, and the best way to regulate all this biological pathway would be regulation at the first stage, which we call transcription. This is answer A. And next question, which of the following statements about an animal bearing a somatic mutation is true? Answer A, some but not all of the animal's offspring will also carry the mutation. This is not true, because somatic mutation means that this mutation happened not in the sex cells, not in the germline that would produce gametes. Male gametes would be sperm, female gametes would be egg cells. So only those mutations that happen in germline cells would affect the following generation, and any other mutations that you may experience your body, say even some people may have a cancer or any other changes, such mutations wouldn't affect the progeny unless they would happen in the sex cells. So answer B, all of the animals' offspring will carry the mutations. This is also not true. Answer C, both the animal and its offspring will show the mutant trait. This is also not true. Answer D, the animal but not its offspring can be affected by the mutation, and this is true. So our choice is answer D. And I also want to give you another example from the plant kingdom. In the plants, for example, imagine that this is a plant, so this is root system, and this is stem, and here we have leaves. And now imagine that all leaves are, say, green, but on one branch you notice that some leaves are variegated, so green and white, or maybe green with white rim. So if you would make a cutting, you would cut this branch, and if you put it in the soil or first in the glass with water, this cutting would give roots, and you would grow a new plant that would have this mutation. And of course every cell would carry this mutation, and also seeds that this plant would produce also would have this mutation. This happens because most of the cells in the plants are totipotent, meaning they can change their function and, for example, may grow new roots or new branch, new stem, new leaves, and so on. But you cannot grow a new animal from its finger, say. Those some animals may regrow their body parts, but basically you cannot grow from cutting of the new animal, unless this is very primitive animal, like, say, worm. So this is one of the differences between animals and plants. And basically, if you want to make your own cultivar, it's not necessary to be a genetist, a selectioner, or breeder. Everything you have to do is just to notice some mutation in plant, take a cutting, and try to grow a new plant. So this is this easy. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.